Hi, everybody. I'm Mike Clegg. Thanks for coming to this Dean's Lecture. And as you probably know, we do these lectures when someone is appointed to the level of professor or promoted to professor. And it's a high bar to get on our faculty, but it's an incredibly high bar to be promoted to professor. And it's not a decision that I or the committee make. It's really the crucial data comes from the evaluation of peers around the world. And so in this case, we're here to celebrate Photini Sinnis's promotion to professor, and it is a special day. And I used to get up and say, I'm really a jaded, cynical person. But the one thing that does impress me was when I got promoted to professor, because it's a big deal, and this is a big deal. And we feel very fortunate that Photini is here at Hopkins and part of our school community, part of the MMI, and part of the Malaria Research Institute. So I'll just tell you a little bit about her, and that's another thing that I do at and I see Arturo Casadevall, who everybody knows, who is the new chair of MMI. Arturo, this is your first departmental dean's lecture, I think, so congratulations. And so what I do now is I introduce the faculty member to people who know her far better than I do, but that's what I'm going to do. And I was a little late because it took me a few minutes to practice Sporozoite to say that a few times. But as you may know, Photini began her career at NYU as a postdoc in the Department of Immunology from 1990 to 91, and then in parasitology. And very early in her career, she made some critical observations of the role of the malarial Sporozoite, and I'll just say CSP in infectivity. And I've heard her speak maybe 3 times, and each time I've been impressed with the clarity of her presentations and also the great movies that she shows. And I know today is going to be a similar show. But she showed that CSP processing was required for hepatocyte invasion, localized the cleavage site, and demonstrated that the signal for proteolysis is in the initial attachment to heparin sulfate in the liver. She became a research assistant professor in parasitology at NYU, and also was an assistant attending physicians in the Department of Medicine at NYU. She rose to the rank of associate professor in those 2 departments, parasitology and medicine, and we were very fortunate in 2011, a relatively short time ago, to be able to recruit her to our school and attract her from New York. She came as an associate professor and then was promoted to professor last year and has a joint appointment in the Department of Medicine and the School of Medicine and actively teaches there as she does here. And then last year, she was appointed the deputy director of the Johns Hopkins Malaria Research Institute. She's been the deputy editor for PLOS Neglected Tropical Diseases and the associate editor of PLOS One. She's been an ad hoc member of the Pathogenic Eukaryotes Study Section at NIH since 2011. She's also been a lecturer and the module director for the Biology of Parasitism course at the MBL since 2011. And I think everybody knows that course and knows MBL. I worked there 2 summers actually, and attended some of those lectures. And it's really the best people in the field are selected to run those courses. She's internationally respected researcher in malaria and in the study of molecular and cellular biology of parasites, malarial parasites. She's done great things, and what's been great for us is that she came with a foundation of doing great things and has built on that while she's been here. And I think that's what she's going to tell us about during her talk today. Just a little more about her background. She went to Dartmouth Medical School. I guess it was medical college and medical school. And in Hanover, New Hampshire, after having come there from Swarthmore, where she got a degree in biology in 1981. She received a Howard Hughes Medical Institute Research Scholarship in 86 to 87, and then she was an AOA at Dartmouth as well. She did her internal medicine residency at Columbia in New York. So Photini, thank you for, as I said, for coming to Hopkins, and we're looking forward to your talk.
Well, thank you for that very nice introduction, and thank you everybody for coming. I will start. I I see many colleagues, many people from my department in the audience who don't need some of this introduction, but I sort of scripted this for a broader audience. So I work on malaria, which is which remains one of the most important infectious diseases in the world. It's caused by protozoan parasites. So these are, one-celled eukaryotes. They're more complicated than bacteria and viruses of the genus Plasmodium. And these parasites are transmitted to humans by Anopheles mosquitoes. There are 5 species of Plasmodium that cause disease in humans, with the most severe disease, with Plasmodium falciparum being the species that causes 98% of the mortality. There are about 500 million cases of malaria a year, and over 800,000 deaths a year due to malaria, and most of these are in children living in sub-Saharan Africa. So I thought I would show you what's happened over the last 150 years in terms of the malaria map on the world. So if you look in the light pink areas and then going to the dark pink, in the late 1800s, all of these areas, in all of these areas of the world, there was malaria transmission. And the only not affected areas are places either where there were no people, where it was absolutely freezing cold for the vast majority of the year, or where or places that had very high altitude, and so mosquitoes don't survive as well there, or where you have deserts. So you can see that there was malaria in temperate regions as well as the tropics and subtropics. So over the course of the last 150 years, and most notably since the end of World War II, there's been a really significant contraction of malaria transmission in the world. And so now it's really in these dark pink areas. And so it's in the tropics and the subtropics of the world where we have malaria transmission. And what happened? Well, on the one hand, I think there's a few things going on here, but I think one thing we have to remember is that in the temperate climates, the force of infection was probably lower than was lower than it is in the tropics and subtropics. So we have winters, and winter is a time when mosquito populations go way, way down. And winters can last several months. We don't, in the summers we have humidity, but there's we don't have as much year-round humidity as they do in the tropics, and mosquito longevity is correlated to humidity. So on the one hand, it's going to be much more difficult to get rid of malaria in these areas, and the interventions that we had that led to the contraction of malaria and the eradication of malaria in the temperate regions of the world are interventions that will do something in these tropical areas, but won't eliminate it as it did in the temperate regions. So one way, there are studies that have actually, experiments that have actually been performed and that tell us this. So the Garki project really tells us how far we can get with current interventions in areas where the force of transmission is much higher than it was in the temperate regions. And the Garki project really ran in the early 70s, and it had 2 pillars. There was residual indoor spraying with insecticides, and there was mass drug administration. And they did this for a few years, and they kept incredibly good records, both before and after the project. And it's one of the best epidemiological studies on malaria control that we've had. And they decreased the prevalence of parasitemia, of parasites in people, by 99%. They decreased morbidity and mortality significantly, but they could not interrupt transmission. And when they stopped mass drug administration and insecticide residual spraying, it took about 3 to 5 years for for transmission to come back full force. And I talk about the Garki project, which was a while ago, because the records were so good and so precise. But really, we have had sort of similar situations in many parts of sub-Saharan Africa more recently, where groups have gone out and done intense treatments and insecticide spraying, and transmission has decreased significantly. And then when these interventions are lapse a little bit or a lot, things come back full force. So if we want to control malaria in the parts of the world where transmission is currently occurring, we really need to add to our armamentarium. And we actually haven't added much to it over the, since over the last several decades. So I would argue that two things could make a difference. One is vaccines, and the other is drugs targeting multiple life cycle stages. And I will go over the malaria life cycle with you, but there's one stage that causes symptoms in people, and all the drugs target that stage because you go to the doctor because you're sick, and so you need to target that stage to make somebody better. And we have had new drugs because the parasite has become resistant to our old drugs. But really getting new drugs that target blood stages is a bit of a Red Queen kind of race. We're just running to stay in the same place. And it's important. It's absolutely critically important. But if we want to win the war, we really need to target other stages. And I think that now there is some recognition that we might be able to target many stages with one drug. So for people who have not seen this before, I have to go over this before I talk about my work, because it's integrally involved in what I do. So malaria infection is initiated when an infected Anopheles mosquito injects sporozoites. As you will see, they're injected into the skin, and they have to reach the bloodstream to go to the liver. They must go to the liver in order to develop into the next life cycle stage. So these sporozoites invade hepatocytes and then divide and have a huge replication rate. One sporozoite will divide into 5,000 to 10,000 individual merozoites. This stage, these are called pre-erythrocytic stages, because as I will show you, not very many sporozoites are inoculated by a mosquito, and not very many of those that are inoculated get out. And so this is a time when parasite numbers are low in the host, and so it's completely asymptomatic. You have no idea that you have been infected. However, once these mature liver stages rupture and release merozoites, they infect the blood. And then in the blood, you have these iterative cycles of replication. And this is responsible for all the clinical symptoms of malaria. And now parasite numbers go through the roof. They're on the level of 200 billion parasites or more. I will also mention that a mosquito has to get infected again, right? So there's two ends of this. The mosquito infects us, and then we infect the mosquito. And some of these blood stage parasites differentiate to sexual stages, which are taken up by a mosquito. And then these develop in the mosquito to ultimately lead to sporozoites. And so that's the cycle. Okay. So in my laboratory, I work on the sporozoite stage. It's this transmission stage. And I just want to go over what sporozoites do. They develop in oocysts on the midgut wall of the mosquito. They go to salivary glands. They're injected into the skin. They enter the blood circulation. they go to the liver and they develop into these liver stages. And we're interested in the sort of cellular and molecular interactions between sporozoites and their mosquito and mammalian hosts. And my talk will be divided into 3 parts. First, I'll talk about the initiation of infection in the skin. And then I will I'll discuss some more molecularly oriented work and talk about the role of 2 proteins in sporozoites getting out of the skin and going to the liver. I also want to say, I have to actually remind you before before I get into my work, that I've talked to you so far about human malaria parasites, but there are other animals that get malaria. So there are rodent malaria parasites, and there are non-human primate malaria parasites, and there are bird malaria parasites. Now, the rodent, primate, and human malaria parasites have identical life cycles and are all transmitted by Anopheles mosquitoes. And we study, much of our work is with the rodent parasites. And so what I will show you, that is what I'm going to show you. In particular, doing work with stages of parasite, the stage of the parasite where there are very few parasites, where numbers are low, is especially difficult to do in humans. With blood stages, you have 200 billion parasites in the blood. You can ask someone for some blood, and it's not that hard to get them to give it to you. Liver biopsies and skin biopsies and things like that are more complicated. But I will also say that the rodent model has given us a lot. All the initial work for what is now the most successful malaria vaccine candidate was initially done in the rodent model. So I'll start with a movie that was not made by me. This was made by a former colleague of mine, Jerry Vanderberg. And before I start the movie, I just want to orient you. So what we're going to do is sort of look with movies at the initial stages of malaria infection in the rodent host, in the mammalian host. So on this slide, a mouse is anesthetized on the microscope stage, and you just see the autofluorescence of its ear. And this is a mosquito proboscis, pseudo-colored red, and the mosquito is infected with fluorescent parasites. And you can see that that as the mosquito is looking for blood, it is spitting into the ear of the mouse. So with its saliva is coming the sporozoites. So all blood-sucking insects have to find blood, and so this is the finding phase. And once they find blood, they imbibe blood. The mosquito, actually, the sporozoites come out the salivary duct, and this duct is completely separate from the duct in the proboscis that sucks the blood. And once the mosquito has located blood, it stops salivating and it sucks. So what you just saw, sporozoites being deposited in the skin, is in general what probably occurs. I would say over 98% of sporozoites are probably inoculated into the skin. Because once that mosquito finds blood, either be it a pool of blood, or sometimes it actually can cannulate a vessel, it stops spitting. There may be a split second that it takes to stop spitting, so there may be some sporozoites that are inoculated IV, but the majority are inoculated into the skin. And this has really only been appreciated through the work of several laboratories, including my own, over the last, I would say, 7 or 8 years. And even still, there are people that don't believe it. So once sporozoites are in the skin, they have to move, and they are capable of a motility called, they are capable of moving, and they move quite fast. And Christine Hopp in my lab made this movie. So this is, again, the ear of a mouse. The mouse has been inoculated with antibodies that label the vessels. So you can see the vessels here in pink. These green things are just autofluorescent hair follicles. And you can see that sporozoites just move around the skin. They look kind of aimless, but and they may be. But when they contact blood vessels, they sometimes know that they have contacted blood vessels. And actually, Christine has found that their movement, she's tracked their movement and analyzed their movement and found that it changes when they get near blood vessels. So they actually do recognize blood vessels. And here's another movie from Christine. Let's see. Where you can see sporozoites entering the blood vessels. So here's a sporozoite, and it's definitely not coming off the vessel. It probably recognizes that it's in the right place, and it takes a little time, and it does seem to take time for this, and then boom, it goes. So sporozoites get inoculated into the skin. They must be able to move in the skin to find blood vessels and get out. Some sporozoites, a proportion actually, also get picked up and go to the lymphatics, but these never make it to the liver. They never actually generate a productive infection. Although, as Fidel has shown, they can prime the immune response. The immune response. So the last movie I'll show is this. It just puts that whole sequence together of inoculation, movement, but you don't really see blood vessel invasion, but you sort of can. I'll show you. So we don't have blood vessels labeled here. This is just autofluorescence of the hair follicles, and this is the mosquito is inoculating as the movie was started being taken, and you can see this. This group of sporozoites, and this is a 2-hour time lapse. This is just 2 minutes. We're not going to sit here for the whole movie. It's a 2-hour time lapse, and what you notice is that their movement, that they disperse over time. And here there's a blood vessel, because if we watch here, we see that sporozoites shooting out of the picture. So they have found it and they have left the scene. And then over time, there's more dispersal and invasion. Okay, so how many sporozoites does a single mosquito inject? Well, to answer that question, a former postdoc, Darcy Medica, did what we called the Van Gogh experiments. She did single mosquito feeds on ears of mice. So one mosquito, one mouse. You cut the ear off after the probing, very shortly after the probing, immediately after, and with quantitative PCR, you count. And she found that the median number of sporozoites inoculated is 18, so it's quite low, and with the mean being 123. And this tells you that the range is huge, and the range is huge, with a favoring towards the low end. So the majority of sporozoites the majority of mosquitoes inject very few sporozoites. We also wanted to know how long they stay there, how long does it take them to get out, and we inoculated sporozoites either with a needle or by mosquito bite. And then we did a slightly different experiment where we took the ear off at different time points and then kept the mouse and later harvested its liver to see if we take off the inoculation site, how does that affect the parasite burden in the liver? And we found that if we removed the ear at 1 hour, we always saw parasites in the liver, but not a lot. And if we took the ear off at 3 hours, there was no difference from controls. So many of the parasites take quite a while to get out of the skin. So the summary thus far is that sporozoites are inoculated into the skin as mosquitoes probe for blood. The median number is low, it's 18. And though there are early immigrants, i.e. sporozoites that leave within the first 5 minutes, the majority take between 1 and 2 hours to exit. And I also just want to point out, although I've indicated that the inoculum is low, I just wanted to sort of emphasize that point with this graph. So The graph mostly focuses on sporozoites, but some of the other stages are included here, and I'll just take you through it. So in the mosquito, there are not very any oocysts. Those are these things on the midgut wall in which sporozoites develop, because the oocyst is the product of the last transmission stage. The mosquito taking up the gametocytes, you have to have fertilization, and then you have to have these O-kinates go through the midgut. And because of that, so this is a bottleneck. This is clearly a bottleneck for the parasite, and it's a bottleneck that many of my colleagues in the Malaria Research Institute study, and I think it's extremely important. But here you have an expansion, though, because in one oocyst, again, thousands of sporozoites develop. And then these are released, and about 10% of them get to the salivary gland, so you have a little decrease. But just because they're in the salivary gland doesn't mean they get inoculated. It's actually only a very small fraction of what's in the glands that gets inoculated. So now, as I told you, the median is, somewhere between 10 and 100 sporozoites are inoculated into the skin, usually on that lower end. But only a fraction of these actually enter the blood vessels. And that's a number that we don't completely know yet and that we are trying to find out. It's actually not the easiest number to determine. But based on the experiments we've done, I think that it's on the order of between 10 and 20%, probably closer to 10%. So again, now the sporozoites that get out of the skin and successfully go to the liver drops again. So the number of liver stage parasites you have, the number of successful sporozoites and liver stage parasites you have is 10, less than 10, 1, 2, very little. And then you have this tremendous expansion again. You have seeding of the blood, and you have an even more tremendous expansion. So clearly, these transmission stages, here where we end up with oocysts in the mosquito, and here where we end up with liver sporozoites in the liver, are real bottlenecks for the parasite. And I would argue that they're great targets. In summary of that part of my talk, that there's a distinct phase of malaria infection that occurs in the skin, and that this is the spark that gives rise to the fire. So now I'll talk about our work on the two most abundant sporozoite proteins, and the first protein I'll talk about is TRAP. It's the thrombospondin related anonymous protein. And this is just a cartoon of its structure. It is a transmembrane domain protein. It has a transmembrane domain so that you can see its orientation in the plasma membrane of the sporozoite is such that this is outside of the sporozoite, and this cytoplasmic domain is inside of the sporozoite. This extracellular part is composed of these cell adhesion domains. And so this is an adhesive protein. It would be predicted to be an adhesive protein. And it was found by Victor Nussenzweig that this, and people in his lab, that this protein was required for movement, that movement that I've been showing you of the parasite. And its role, this very simple cartoon depicts its role in the motility. So here, just to orient you, this is outside of the sporozoite. This is a substrate. It could be the skin. It could be or a glass cover slip. And the extracellular domains are binding here. And the transmembrane domain is here, and the cytoplasmic tail is binding to the motor, which I have not shown in its complexity at all. And this is linked now to an intermembrane complex. And the way the sporozoite moves is that this motor translocates this protein bound to substrate posteriorly, and so then the parasite moves forward. And I thought, just to sort of help explain how that happens, you can think of a swimmer, right? So the swimmer is pushing the water posteriorly, and then in that way moving himself or herself moving forward. But the sporozoite is a little different from the swimmer in that, It doesn't have arms that can sort of flap like this and push substrate back. It has this protein that's relatively fixed in the membrane. The membrane is fluid and it's going back, but then what happens to it? Right? It's linked here to the substrate and linked to the motor. It's being translocated posteriorly, but for the parasite to keep moving, this has to release at some point. So we hypothesize that has to happen, and that likely that this release occurs via the action of a protease. And to look at whether trap is released, I thought I would just show you one of the many ways that we found that it was released. So this is just sporozoites moving on a cover slip, on a glass cover slip, and they move in circles. In vivo, they don't move in circles as much. And if we can fix them we can fix them at any time, and then stain, and we see that, and we're staining for trap here, we can see that trap is left in the trails. So that suggested to us that it is indeed shed from the parasite. And our hypothesis as to what is doing the shedding is that it was a protease, it's a member of a family of proteases that are in membranes and cleave proteins that intramembranously. So this family is the family of rhomboid proteases. They have 7 transmembrane domains. This portion is catalytically active and cleaves its substrate within the membrane. And we had this hypothesis because if you look at the transmembrane domain of trap, you see a rhomboid cleavage site. And so to determine whether that was the case, we made mutants, where we replaced the endogenous copy of the protein with a transmembrane domain in which that cleavage site was mutated. And if you look, I just showed you this movie, so you don't have to see that again. And this is our mutant. And we call it our constipated mutant. It really, it gets stuck all the time. So it gets stuck and then it moves forward. So something is ultimately cleaving trap, but it's not this smooth, fast gliding. So these guys go very slowly. When we did infection studies with those parasites, we did infection studies, and this is how we did them. We basically took either our wild type or our mutant parasites, and we either injected them intravenously or we injected them into the skin, intradermally, which is how a mosquito injects them. And then we monitored the mice for ultimate blood stage infection, right? So if none of those parasites get out, you're never going to get a blood stage infection. And the mice inoculated with control parasites, either intravenously or intradermally, all became infected. The mice inoculated with the trap mutants intravenously became infected, but the ones that were inoculated intradermally never became infected. And so this tells us that, again, it sort of supports the idea that gliding motility is extremely important for getting out of the skin, and not only that, that you have to go fast. And Christine has actually imaged these mutants in the skin, and this is a star plot of, so it's a maximum projection of the tracks of each sporozoite in a given movie, and each sporozoite is colored differently. But then you take all the tracks and you put them in the center because it's prettier that way, and also I think it gives you some information. It's easier to get the information, actually, than in just a maximum projection. So you can see this is a star plot of wild-type parasites in the ear of a mouse, and this is a star plot of our trap mutant. So it's slow and it's not moving as far. And when we look at how far it moves and extrapolate that, these mutants have about a 90% reduction in the volume of tissue that they explore. And consequently, they have the much less likelihood of actually finding a blood vessel. So Christine painstakingly quantified blood vessel invasion, and each dot here is a movie. And so she looked at the percent in each movie of sporozoites that find blood vessels. And this mutant, there was only one blood vessel invasion event in one movie. So it's not very good at getting out. Sporozoites that cannot efficiently remove trap from their surface move slowly and in turn cannot efficiently exit the skin. And that really highlights the importance of gliding motility and fast gliding motility in the infection process. We've all, now I'm going to just talk a little bit about, in rather broad strokes, our work on the circumsporozoite protein. As many of you know, but not all of you know, it's the major surface protein of the sporozoite and really forms a dense coat on the parasite surface. And it has this cell adhesion domain in the carboxy terminus, and that is conserved among all species of Plasmodium, human, primate, rodent. And it also has a conserved 5 amino acid sequence here, N-terminus to the repeats. And this protein is the basis of the only malaria vaccine in phase 3 clinical trials. So it's actually an important protein, and it's something that we should try to understand how it functions. In order to dissect how it functions, we made antisera to the different regions of the protein, and we found that this cell adhesion domain was only exposed at the two ends of the sporozoite's life. And in the middle, the cell adhesion domain appeared to be masked. So it's not that it's not there. If you permeabilize the sporozoites or make their surface, loosen it up a bit, antibodies will get in. But if you just take antibodies and you just lightly fix the parasites or do it with live parasites, antibodies to this region do not recognize these migrating parasites. And so we actually don't know how the structure of this protein, and it's an active area of investigation in my laboratory, but we think that this is one possibility where the cell adhesion domain is here. It's lipid anchored onto the parasite surface. It's not a transmembrane domain protein. It's anchored with a lipid tail. And this is, let's just say, this is the surface of the sporozoite, right? So it's possible that it's just densely packed like this, and so that this N-terminus does not allow for antibodies to get through. The other possibility is that this is somehow interacting and actively masking this cell adhesion domain. We don't know. And this repeat region is actually thought to be somewhat flexible. And so, okay. So how does this become exposed when the sporozoites enter the liver? We found that there's a parasite cysteine protease that clips in this 5 amino acid sequence that's highly conserved and removes the N-terminus and leaves us with this that is accessible to, where antibodies can recognize now the surface cell adhesion domain. So we don't know exactly what the function of cleavage is, but some clues were. Actually, before that, one more slide. If we inhibit cleavage we can actually inject mice with cysteine protease inhibitors. They're remarkably not toxic for mice. And if we inject mice with cysteine protease inhibitors and so inhibit this cleavage event, and then inoculate parasites, this is the protease inhibitor injected mice. These are the control mice. We can significantly reduce infection. So to look at the function of cleavage, we actually made a mutant in which only the cleaved form of the protein is expressed on the surface of the parasite. And we found that these parasites get stuck in the skin. And Christine has imaged these parasites, and these are star plots once again. And this is our wild type. This is the same one I showed you before. And you can see that this mutant actually moves in the skin much more than the trap mutant. And when we look at it in vitro, its speed is normal, and it moves pretty normally. But when she looks at blood vessel invasion, it's again, with this mutant, it's a very rare event. So we're not absolutely sure, again, in this case, we're not absolutely sure why they're getting stuck in the skin. We don't know yet what the cell adhesion domain is binding to. It may bind to blood vessels. It may be important, or it may be binding to something non-specifically in the skin because it is an adhesion domain. And these are things that we're actively trying to figure out. So just in conclusion of this part, the adhesion domain of CSP is masked as sporozoites migrate from mosquito to mammalian liver, and cleavage of the N-terminus exposes the adhesion domain and is associated with the switch to an invasive sporozoite. And premature exposure leads to sporozoite arrest in the liver. So in summary of all the sort of data I showed you, I think we can say that the skin phase of malaria infection is a bottleneck for the parasite, that motility is required for sporozoite exit from the dermis, and that parasite proteases regulate sporozoite adhesion. It regulates, in the case of trap, removal of trap from the surface by a rhomboid protease disengages adhesive interactions and enables motility. And in the case of CSP, removal of the N-terminus exposes an adhesive domain and enables the parasite to switch to an invasive mode. So why do all this? Well, as my colleague across the street, Ron Schnarr, says, biological knowledge provides therapeutic opportunities. So how can we use this knowledge to intervene? Well, I think the first thing is that we should be thinking about targeting sporozoites in the skin. Now, while it's true it's going to be difficult to get 100% of them, I think if you significantly decrease the inoculum, you can really impact transmission, because transmission is a little bit of an iffy thing to begin with. And that motility is also a great target because it's so essential to the sporozoite for infection. And we've been working some with Jürgen Bosch. Jürgen's been designing drugs to target the motility apparatus, and we've been looking at the effects of his compounds in the skin. And I think that targeting the proteases and targeting possibly the cleavage sites with antibodies may also be valuable or productive. And so I just want to end with 3 more slides talking about some recent work that we did and that Fidel and his student Diego Espinosa did. So to just introduce that work, I'll say that I'll talk. RTSS is the most advanced malaria vaccine candidate, and I alluded to this earlier in my talk. RTSS is this part of CSP fused to the hepatitis B surface antigen and administered with some pretty special adjuvants, and it has shown about 50% efficacy in phase 3 clinical trials. Because of our work, as you can see, the cleavage site is not included in RTSS. And I think that the demonstration that this is a functional domain caught the interest of some people. And the NIH funded SAIC to make antibodies to the N-terminus. And one of the antibodies they made bound right here, right next to the cleavage site. So we actually looked at whether, and this is with falciparum now, because of course that's what they're interested in. So using falciparum sporozoites, we found that this antibody actually inhibits cleavage of the protein. And I don't have to go into detail, but this is just a an experiment showing that so this is the full length here, and then this is the cleaved form here. And normally we can feed parasites radioactive amino acids, and we see that initially the protein is here, and then it gets clipped, and you can see the bottom band. And if you do this experiment in the presence of this antibody, you have very little cleavage. So Diego in Fidel's lab actually looked at the effect of the antibody on infection in vivo. And of course, we did this with mice, but Diego has parasites that express falciparum CSP. And he inoculated the antibody and then allowed infected mosquitoes to feed on the mice, and then harvested the livers to see if there was an effect of this antibody. And this antibody actually inhibited liver stage parasite burden compared to naive controls. And monoclonal 2A10 is an antibody that actually recognizes the repeat region, and that has inhibitory activity at high concentrations. And so this antibody works as well as this repeat region antibody, but together they synergize and work better. So that's interesting, and possibly and PathMVI is interested in potentially sort of pursuing this line of work, and possibly broadening our antibody repertoire against CSP may be beneficial in terms of the vaccine effort. So I just want to acknowledge the people in my lab, past and present, and they did all the work that I showed you. And the funding that I've had over these past years has been from the NIH, PathMVI, the Gates Foundation, and the Bloomberg Family Foundation. Thank you.
Thanks, Photini. It's neat to see how your research has progressed. And the first movie I ever saw you was one where you showed inoculation, cannulation of the blood vessel by the mosquito, and injecting straight in. So the idea of the pathogenesis has changed a lot. So it's neat to see. Well, I shouldn't say we can talk later. But let me ask: any questions, or Arturo?
Photini, that's a very nice talk. You cover a lot of material very short. I'm struck on the motility patterns, in that there seems to be, to me, two patterns. Mm-hmm. There seems to be the ones who appear to have like random walks and are tighter, and the ones who branch out. Are these two phenotypic variants?
So we have actually looked at the motility patterns of these parasites. And usually when you see a parasite going like this, in a more constrained fashion, you think, you know what, there's a blood vessel nearby. And Christine has actually looked at motility of sporozoites on and near vessels and in the avascular portions. And there's a clear difference. And so the mean square displacement of parasites, of sporozoites, that are not on vessels or near vessels is much higher. So the mean square displacement goes down when they're near vessels. So they're doing more circling. They're still moving. They're traveling, but they're not moving distance. And their speed goes down.
So that would suggest that they're sensing something in
Absolutely.
the environment, probably an oxygen gradient or a CO2 gradient. Have you thought of taking your mice and putting them in a high oxygen atmosphere?
No.
And see if you can change the pattern?
That's a great idea. Yeah, so we were thinking that this suggests a recognition event, and we were going to test, all the suspects, but we hadn't thought about oxygen. So that's, yeah.
Yes, that's easy to do. You
Yeah.
just take your mouse
and see if it changes.
and see if you see the pattern change. That'd be
Yeah.
very nice.
Thank you. Other comments or questions?
my voice is just not loud enough. Okay, Photini, I was like wondering, just like from a perspective being not really actually a eukaryotic person, so a tissue like the skin appears to me an extremely tough, actually three-dimensional structure where you move. So I'm a little bit wondered what your idea is about actually what provides the force for these parasites to squeeze themselves through that, because I think they encounter, I mean, there's not really an empty space. When I look at these movies, it looks like as if they're effortlessly gliding there through something which is like a surface of a glass slide.
Right.
But I think, I mean, from my perspective, it's kind of like you're moving through a jungle of cells which are very closely, tightly knit together. So actually what provides the force? Really only the shedding of these membrane-associated trap proteins, or do you think there are other mechanisms that push these cells through that very dense matrix?
So there aren't a lot of cells in the dermis. There are some, and it will encounter some cells, and it does. Sporozoites, which I didn't talk about, have the capacity to go through cells, right? That's a different. But going through matrix, the vast majority of what's in the dermis is extracellular matrix, and going through that, I've thought about this a lot, right? So we would like to look at whether the parasite is secreting metalloproteases. We've recently done a sporozoite surface proteome, and there is a metalloprotease that's expressed on the surface. So that's, so one possibility is it's making it easier for itself. It's certainly moving in a helix, and so it's corkscrewing, which is hard. You can't really appreciate because it's moving 2D. So quickly that, and the movie's 2D. Yeah. And the models on motility are, people are sort of wondering, is there more than what I showed you?
So did somebody else have their hand? Yeah. Gary? If you use a microphone because it's being recorded. That's what I understand. Yeah.
So I wonder, if you're looking at antibodies against TRAP and CS for immunization purposes, are there antibodies in the dermis? And if so, how do they get there? And if not, how do you get them there?
So that's a good question. In fact, I was just discussing that with people in my lab. We can inoculate antibodies IV, and I didn't show this data, but then look at sporozoite motility in the skin. And if we inject a high concentration of what we know to be an inhibitory antibody on motility, we stop them in the skin. So now how did that antibody get there? Was it transcellular, things can go from the luminal surface of the endothelial cell to the abluminal surface through endocytosis. And it's possible that they get there that way. Or when the mosquito probes, you break blood vessels and you're gonna, and that's actually, we don't know. And I've been trying to find a literature on this. I talked to doctors, I talked to immunologists, this is a black box.
The volume of distribution of IgG, it will extravasate.
It will extravasate through...
Normally, yeah. And also once you set up an infection, you're going to have leakage.
Exactly.
Within minutes.
Right.
So how is the vaccine given? Is it given intradermally or?
No. I am.
No. And would it make any difference if you gave it intradermally?
I don't think so. But actually, you raise a good point in that. One thing I forgot to say, it looks now from immunized people that the vaccine is largely acting through antibodies, which may largely be acting in the skin.
I had a different question. So regarding your last 3 slides with Fidel's and Diego's work, if you look at the monoclonal antibody that's specific against 3D7, probably Plasmodium, have you looked at the variability, what is existing? Like if you look at PlasmaDB on the genome side, the region that's recognized by that antibody, is that variable or is that conserved in all isolates which you can get? And would that be feasible to use this as a, let's say, subunit vaccine then?
Right. Well, so region 1, which is the cleavage site, is conserved in all parasites. Upstream is not conserved, and that's where this antibody is binding. But they didn't make a region 1 they set out to they made monoclonals against the N-terminus, right? And then they had this one, and they gave it to Fidel and to me. So it's sort of a proof of principle, but I think the idea would be to make a cleavage site, to make the cleavage site, as an immunogen, because that is conserved.
So if there are no burning questions, or even if there are, maybe we can do it outside at the reception because I know people have to leave. But I want to thank you for a great talk.
Thank you.